part one. Isn't he a beauty? All right. So, we'll start with the function. So obviously, reproduction of the offspring is very important function of reproductive system. And then development and maintaining secondary sex characteristics such as voice and hair presentations and more. We'll talk about that. So reproductive period, as you all know, begins in puberty, and we have several kind of stages of the process. Some of you are probably familiar with biology classes, YouTube. The term gametogenesis refers to formation of the gametes, right? And the gametes, what are the gametes? That's a collective term for what two main cells?
Sperm.
The egg and the sperm, right? Or egg is what we call it ovum, right? So gametes is a collective term. So gametes both, the sperm and the ovum. When they fuse, when the eggs get fertilized, then, so the process called fertilization, they will form something called a zygote. So that zygote is actually that first cell where the egg with its own chromosomal tissue set up, and the chromosomal information, DNA information from the sperm come together forming the zygote. Zygote at this point has two pairs of the chromosomes because the gametes, sperm and the ovum, will only have a single pairs of the chromosomes. And so the zygote is that first formation. And after that, the zygote will divide via mitosis. If you guys remember, mitosis is the production of two identical cells. So the zygote being this one cell, and then it divides in two, and each one of these two divides in two, and each one of these two divides in two, and they carry the same genetic information. At this point, all cells are identical genetically, and you may be thinking like, well, how's all these cells that are being mitotically divided identical genetically, but then how do you get all these different tissues that you're going to see in the grown baby, right? Well, every single cell of our body has the same chromosomal setup, but not all genes are expressed. So the pancreas will express certain genes and silence others, and your brain cells, neurons will have certain cells, certain gene expressed, but certain genes will be silenced. But they're all there. They're just not all activated and actually producing the product of the gene. So we use the term an embryo for 0 to 8 weeks of development. So that's an embryo, we call it an embryo. And then after that, we call it fetus, until birth. So embryo up to 8 weeks, right? And fetus after that. That's the terms that we agree to use. So, sex determination. What happened to my little ruler? Oh, here it is. So what this figure is showing to you only sex chromosomes, right? So human has how many chromosomes? Who remembers? How many chromosomes do we carry?
Michael Jordan.
23.
23 pairs,
Yeah,
or
46.
46 chromosomes. Can somebody explain why they disappear every time? I don't understand this. Okay, this is crazy. All right, so 46 chromosomes, 23 pairs, right? So when I say pairs, do you understand that the 23 chromosome came from mom and 23 chromosome came from dad, right? So here, they only show you the sex chromosome, right? So the 22 others are not shown here just for simplicity. So there are 22 others in that female parent. So XX would means that the sex chromosome, that last pair, is going to be X from the mom and X from the dad, right? And the XY would be a male parent, right? X from the mom and Y from his dad, right? So, and of course, since during the gamete development, when the cell divides, right? So the female parent can only send X chromosome to one of the X or the other X. There's no other choices, right? And then for the male parent, it could be X or Y. And so if obviously X sperm fertilizes the egg, we're gonna have the female offspring. If the Y sperm fertilizes the egg, we're going to have a male offspring. This is the most kind of a typical way how that goes, but we'll talk later about possible aberrations and abnormalities or alternative ways to send the chromosomes around. But this is the most frequently seen. So when you look at this is called karyotype. The karyotype is the way to look at the chromosomes from the embryo or fetus and then kind of line them in pairs, and the geneticists will do that because they kind of can identify them by size. When we number the chromosomes in the human body, we number them from we call one as the largest chromosome, and then we just go down the list. So the numeration size. So as you can see, they kind of decrease in size. When you get to 22, they all kind of get much smaller comparatively to the one, right? So it's like one chromosome number one from mom, one chromosome number one from dad, two from mom, two from dad, so forth. And then here's the sex chromosome. They're not really true pair because as you can see, they're quite different, like in this particular example. So this way to display the chromosomal setup of the embryo or fetus is called karyotyping. And now this is a karyotype of what, a boy or girl? A boy, because you can clearly see Y, right? So XY. You also probably notice the difference how large the X chromosome is and how small the Y chromosome is. X chromosome not only carries sex differentiated genes, it also carries a lot of other genes that the embryo cannot survive without the X chromosome. Embryo can survive without the Y chromosome. Embryo can survive with one X chromosome, but not with one Y chromosome. So X chromosome is a must because it has genes for other functions, very important functions. So Y chromosome will be very important, even though it's small, it has a very important genes for the male reproductive structure. We'll talk about that more, of course. So here's the possibility. For example, if you have double X, and the reason that could happen is when this is supposed to split and send each X chromosome to a different egg during the meiosis, it's possible that it didn't split and that the pair was sent to the one egg, right? And so then the karyotype of this offspring would be XX and the Y from the dad, but it's still a male, even though it has two X chromosomes because it has a Y and and that person will survive. Now, if you did not get an X, and we will designate it as the Y0 or YO, some people say that would not be okay. So that embryo will self-abort. You will really never know about that. Because, we generate a lot of zygotes, and they may not ever come to fruition. Okay, so just make sure that we use the terminology correctly. All you guys, your generation is much better at that than before. But just to be sure we're on the same page, sex is what assigned at birth, and it's kind of the biological status associated primarily with the physical attributes, such as do we have a Y chromosome, do we not have a Y chromosome, what's the hormonal display looks like, what is the external and internal anatomy, and so that's usually is a person who's actually delivering the baby that assigns the sex. Gender refers to the roles, the behaviors, right? So meaning like, the women should act this way, and it's different culturally, or males should be doing that. So it is really, the gender is very different culturally, while the biological sex is more similar, because it's kind of determined to best of our medical understanding what that is as a sex. And the gender identity refers to a person's internal sense of being, right? And so what do they feel like, how they want to be expressing themselves, behavior, hairstyle, and so forth, clothes. And then sexual orientation specifically refers to more of the romantic and physical and emotional attraction to another person. So as you can see, that is how the current terminology is used. There is a group of conditions that's called intersex. It used to be called hermaphroditism, and that term is kind of being phased out in the literature. Intersex is what's currently used. When there is a discrepancy between external genitalia and the internal genitalia. So, and then we would use like these different characteristics. Like if, for example, the chromosomally it is XX, but maybe the external genitalia are more towards maleness, then we will just define that condition is the 46, meaning there are still 46 chromosomes, there is no like abnormality in the count, XX and then intersex. And then same with the 46XY intersex, true gonadal intersex, or complex or undetermined intersex. So we'll go over them. So the XX intersex, think about that. So the chromosomes are of a woman, right? Could be ovary of a woman, but the external genitalia may appear male. And that's usually, I mean, there are many reasons for that, but it often happens with the excess of male hormone before birth. As the embryo develops, the excess of testosterone and the derivative of testosterone is high in the milieu and could sway the external genitalia in the direction of the maleness. So one of the possible causes that we've seen in literature is adrenal hyperplasia, where body produces more androgens, and then that could cause male characteristic appear early or not correctly, or if maybe male hormones were taken, like a bodybuilders or other athletes, and that could also sway. There are some ovarian tumors that could produce also similar kind of effect. So usually it is the excess of the male hormones during the embryonic development that result in XX intersex, female chromosomes with the external genitalia that look more towards like a male. And so oppositely to that would be chromosomal XY. And so the chromosomal of the man, but the external genitalia are not completely formed or could be ambiguous, or sometimes it could be clearly female. There is definitely a spectrum on that. And so many reasons for that. One of them is like, for example, the abnormal testosterone formation, abnormality, and the enzymes that actually make the testosterone from the cholesterol precursors. You guys don't really like read it once and be aware and just kind of understand that if testes, for example, not formed properly, not enough testosterone, or something hinders the formation of testosterone, that could kind of create embryonic development environment that kind of sways more into the female external genitalia. Now these two I will not really ask you for, but I just still put that for reference because it is the two others that could also be part of the intersex. And so you could just read that once. We're not gonna really focus too much on that. Okay, does anybody remember term diploid? What does that mean to you? Diploid. All right. Okay, Sam, you gonna try?
Isn't it two?
Isn't two? Two what? Sarah?
Two forms? Or I don't know. That's what I'm thinking. Two forms.
Two chromosomes? Two sets of chromosomes?
Yes, the chromosomes comes in sets of two. each chromosome, like we saw in the karyotype, right? 2 chromosomes 1, 2 chromosomes 2, 2 chromosomes 3, so forth. So here that's how it goes, right? So we have our, in our grown-up bodies, like every single cell is diploid. Every single cell is diploid, except when we form the gametes, we form it via meiosis. What is it? Meiosis, here you go, this way. So every cell is diploid. However, when we form in testes or in ovaries our gonads, the way that, if you guys remember meiosis a little bit, it's a 2 subsequent division, but only 2 times we replicate the DNA, so we reduce it by half. So the gametes called haploid, meaning they only have 1 set of chromosome, only 1 set. And that's good, because once they fertilize, right, zygote will be back diploid. So, because otherwise we will be what, tetraploid. So the egg has, N identifies the haploid cells. So diploid means that 2N is 46, meaning all the chromosomes is N is 23 for human. So 2N is equal 26. But the haploid, it just means only 1 set. So 23 individual chromosomes, not in pairs. And so our egg and the sperm, these 2 cells are both haploid. So the haploid, they produce by mitosis, which is a division where it reduces the number of chromosomes. And then, and as I said, it's okay, because once they merge together via fertilization, we are back to be diploid, and from that point forward, after zygote formation, it's all the mitotic division till we have a babies, and all the regeneration of the cells, and every single body is mitosis, and it's diploid, except gametes. Gametes are the only 2 types of cells that actually haploid, only have 1 set of chromosomes. Under microscope, quite noticeably, when you look at the female cells, you could sometimes see this kind of dense area in the nucleus. So this dense area is called Barr body. Where is it? Here you go. Barr body. And what that is if you're a female, you have 2 X chromosomes, and they have obviously alternative version, but same genes, right? So one from your dad and one from your mom, but the gene lineup is the same because it's the same X chromosome. You could have different variants of that same gene, right? But it is the same type of gene. So what the body does very efficiently is that it decides to kind of make one of the X chromosome not active. And it does it by condensing the DNA to such degree that nothing could be read off it, like nothing could be, there's no transcription, translation takes place. So the genes are there, but they're not expressed. Like you have genes, but they're not being expressed, meaning the mRNA is not made from them, the protein is not made from them. And so one chromosome is active, and another one gets very tight, and becomes inactivated. And so, and it could be either one, paternal or maternal. There's so much research, they're trying to figure out what influences which chromosome is gonna go to sleep and become inactive, but it could be either one. It seems to be quite random, so either one. So that's why sometimes, people could have more of the features of the, the father or the mother, and then sometimes they can understand why the other siblings don't seem to have, because it is a really random process, which chromosomes gets passed on to the egg or the sperm, and then which chromosome is actually gonna get inactivated. So even if you have it, you don't really express any genes from it. Okay, now this is a very important part, definitely gonna get a lot of questions from that. So this is our Y chromosome. So, it has this sex-determinant region that's called SRY gene. SRY gene, sex-determinant region, Y, SRY gene. It's only found on the Y chromosome. These genes are not present on the X chromosome. And their function is for the embryonic tissue, we're gonna talk about in a minute, called gonadal medulla, to become testis. So without SRY gene, that would be any testis, right? And then if there is no gene present, meaning there's only X and X, then the bipotential gonad will become ovary. I'll talk about bipotential gonad in a minute. And then this tissue, embryonic tissue, will become an ovary. So for now, what we need to memorize is that the SRY gene is the gene on the Y chromosome that is make it possible for the testis development. In the absence of it, the embryonic tissue will develop into ovaries, right? So look at this. Up to 6 weeks of fetus, that is the stage that is called bipotential. That means that the female fetus and male fetus are not different. And we have this embryonic tissue that we don't have that as an adult. So here's kidney, but here's this embryonic tissue. This part of it, in blue here, Mullerian duct. This one is we call gonad tissue, bipotential, and this yellow one is Wolffian duct. So this is the embryonic tissue, not present in a grown person. But if female, the gonad cortex, the cortex of this bipotential gonad will become an ovary, and medulla, which is you can't see, but it's in the middle of this gonad portion, will kind of just regress. But if male, meaning has the SRY gene on the Y chromosome, yes, because that's prerequisite. The cortex, which is kind of outside here, is going to regress, and the medulla will form the testis. So it's one or the other. And then, if male, no SRY gene, then the Wolffian duct, this yellow guy, will regress. But if male, meaning the SRY is present, Wolffian duct will form epididymis, vas deferens, and seminal vesicle, and that would only be possible with the sex-determinant chromosome build. Right. And then the Mullerian duct, this one in blue, will then regress. If no SRY gene, Mullerian duct will become fallopian tube, uterus, cervix, and upper half of the vagina. So as you can see, 6 weeks is where that difference start shifting, the development of the external and internal genitalia in the different directions, right? So SRY gene is necessary for development of the Wolffian duct and the forming of the testis. Very, very important. So we'll have an opportunity to look at all the cells in a minute. For now, just kind of thinking about it. The testes will secrete many different hormones once they start differentiating. And they will, these hormones will influence then external and internal genitalia development. There's a special cells that's going to, in testes, that's called Sertoli cells, and they're gonna produce something called anti-Mullerian hormone, or AMH. So that anti-Mullerian hormone will make the Mullerian duct regress. So that's not gonna go in the female direction. And then the Leydig cells will produce testosterone and dihydrotestosterone, so they both considered to be androgens. And testosterone will be very important in converting Wolffian duct into their respective tissues. And then DHT will be actually more important in the male external genitalia differentiation, more important than testosterone actually in the male external genitalia. So see, as testes produce all that, this embryo goes into development as a male. Okay, seems to be a little repetitious. I don't know why I was thinking I wanted that. Let's see. Yeah. Anti-Mullerian hormone, Muller index. Okay. So then, if we don't have SRY genes, if we don't have testis development, then we don't have anti-Mullerian hormone, right? No anti-Mullerian hormone from Sertoli cells, then the Mullerian duct will become fallopian tubes, uterus, and upper half of vagina, right? No DHT from the Leydig cells will develop the female's genitalia. This one's gonna get in a little bit like further past the 6, so this is a 10 weeks male, so obviously we have CRY gene, medulla goes into the testis create the Mullerian hormone, here's the Mullerian duct being regressing, right? And then the cells that the testis produce testosterone, then converting the Wolffian duct into seminal vesicles, vas deferent, the epididymis, and so the prostate development also under influence of the DHT. So this would be the progression with the SRY gene present, and then no SRY present, then the cortex become the ovaries. The Mullerian ducts, of course, is not degenerating because it becomes the fallopian tubes, uterus, upper part of vagina, and so that is the going into development of the female fetus. Okay, the pictures are the same. I think I was just trying to. Sertoli cells, Leydig cells, DHT, yeah, it's all I think I'm just using the same picture, but I wanna finish off all the cells and hormones. So for the Sertoli cells, Leydig cells, anti-Mullerian hormone, DHT, testosterone, you guys are really expected to know that, so just keep driving this, but make sure that you make good notes for that. Okay. So shifting gears a little bit about gametogenesis, so production of the ovum and the sperm, right? So let's look at the females first. So first of all, the cells that we call germ cells, before the egg is actually made, the germ cells, they multiply by simple mitosis. It's not a meiosis yet, it's not a reductive division yet. And so during the embryonic development, as you can see for the female, it's quite different from the male, is during the embryonic development, we have mitotic division to increase the progenitors of the ovum, they call oogonia. And then all these cells are still diploid, so you can see that the chromosome, they simplistically just showing 2. And then we actually start having replication of DNA, and these preliminary oocytes are still in embryonic development. And then that's kind of interesting, is because the DNA replicates, but there is no cell divisions, they don't divide. So then they have to wait to puberty to actually split into 2 cells. And this is going to be our secondary oocytes, not the ovum yet, and the first polar body. When you taught biology, when you listen to biology and maybe in the biology like preliminary classes, they will keep saying how you get 4 cells from the primary oocytes, but in reality, in the female body, it's actually just only 1. Because the first division produced what we call polar body that actually disintegrates. And then that secondary oocytes gets released during ovulation, and it's neat ovulation, fertilization to finish the division, and they will produce another egg and the second polar body, and that second polar body will integrate. But really, so it's only gonna be one egg produced from the primary oocyte. But it is true for the sperm that the primary spermatocytes will do a first division and a second division, and will produce 4 spermatid that they will maturate into the sperm by, kind of increasing. But there's some anatomical changes with the flagellum growing and all that, but it is 4 cells out of the primary spermatocytes. But the oogenesis is only produced 1 ovum from the primary oocyte. So what happens sometimes we call that the mistakes of meiosis, it's called non-disjunction. Non-disjunction means that the replicated chromosomes, we call it sister chromatids, they had to descend to 2 different daughter cells, but sometimes they don't. So here's an example. This is for simplicity, they're showing you 1, 2, 3, 4 replicated. You can tell they're replicated because they join it here at the centromere. So 1, 2, 3, 4 replicated chromosomes. And then this chromosome should have be on this pole of the cell so it could descend to these daughter cells. So during the meiosis 1, so there is, in this example, there was non-disjunction. This second pair of the chromosome did not separate. And so now we have this daughter cells that has, see this one's separated. This one's separated normally, but this one did not. So we have the first chromosome and 2 of the same chromosome from here. So then we say, and then if the normal meiosis 2 separation was normal, like this did not carry again together, and there's no non-disjunction in the second division, then you will have n+1. So there's 1 extra chromosome in the final daughter cells, right? In the final gamete. Now this cell obviously will be lacking the chromosome because none of the second chromosome actually went into that, and that cell will self-abort because obviously we don't have all the sets of the gene. That's not gonna work. Now these gametes, if fertilized, potentially could become an embryo, and it has to do with like which exactly chromosome was non-disjunction. So you have an extra one, right? And they're compatible with life. Like, of course, you guys all aware of like the trisomy 21 for the Down syndrome. Here's an example where the first division went normally, but the second division had the non-disjunction. So regardless, it happened in the first division or the second division during the gamete formation, -1 chromosome will not survive, but the +1 might survive, the embryo might survive. So like the trisomy 19, trisomy 21, which is a Down syndrome, right? So the word trisomy says that the chromosome 21, you have 3 copies of it instead of 2, or trisomy 19, you have 3 chromosome 19, and they're compatible with life, and so is a few others. So I think the hormonal control gonna get a little busy, so we're gonna stop for now because we have 2 more lecture periods. It's fine, we have plenty of time. So I'm not doing the question of the day on Thursday, but we're gonna continue with chapter 26, okay? Please sign your name as the page travels. Okay, hormonal control of reproduction. So back in the day when we talked about endocrine system, we obviously did mention the gonadotropin releasing hormone, right? Mm-hmm. Gonadotropin releasing hormone is produced by what? Hypothalamus or anteropituitary gland? Hypothalamus. And it affects the release of the LH, which is luteinizing hormone, and FSH, follicular stimulating hormone. So between LH and FSH, there is what we call short loop negative feedback. Short really meaning just because of the anatomical location. Meaning like when they low, this will be high, and when they high, this will be low, right? Because remember negative, positive feedback? But the long loop feedback, the one from the gonads, is actually it's not straightforward. Specifically, low estrogen or androgen, so androgen would be like a testosterone, DHT. So low estrogen and androgen will have the, obviously, the no negative feedback, absence of negative feedback, and the gonadotropin will increase. The moderate number is actually will exert negative feedback, and it will decrease. So this is something that you wouldn't expect, right? And then the high androgen does also have a negative feedback, but look at the sustained high estrogen. It actually gives the positive feedback and increases the gonadotropin. Something you wouldn't expect, like why is the estrogen is high and the gonadotropin is high as well. So, but it'll come more clear when we go through the female reproductive system. Okay, so first of all, just to remind you, androgens, testosterone, DHT, estrogen, progesterone produced by both sexes, right? Both sexes produce that. And then androgen does have a higher production in male, and estrogen dominates in female. The gonadotropin hormone, as we just mentioned, secreted by hypothalamus and target anterior pituitary gland for release of LH and FSH from the anterior pituitary, right? The role of the luteinizing hormone is stimulates sex hormone production. If we can go back for a minute. So luteinizing hormones play a very important role in the production of the hormones like estrogen, androgens, testosterone, estradiol. It also, only in females, has additional effect on the gamete production. But the FSH mostly has an effect on gamete production. FSH mostly gamete production. There is also what we called here peptide hormones. Inhibin and activin, they not only produce in gonads, they're produced by many different parts of the body, even in the pancreas and placenta and mammary gland, but specifically in gametes, they also have a very important role. We're going to mention them. The inhibin, as the name suggests, blocks gametogenesis, meaning production of the gametes, and the activin stimulate production of the gametes. Is the piece of paper traveling? Where is it? Okay. I just need to take it back. Don't forget. Don't forget to give it back. Okay. When we talk about sex characteristics, we differentiate primary and the secondary sex characteristics. So in the primary, we usually mean internal organs, external genitalia, and how the males and females distinguish. But then on the secondary, more like the bone and the muscles and the anatomy of the hips, the facial hair, or anybody hair, how thick of the voice, sex drive, that is considered to be secondary sex characteristic. This is kind of partially what we already have, but it has a little extra information. So first of all, gonadotropin release, what's interesting, it does have this effect on LH and FSH, and it actually tonic. Maybe better use the word pulsative here, because it is actually releases every few hours, hour and a half, I think, hour and a half peak. So, but it releases before that. So it's kind of continually pulsatively affect the anterior pituitary gland. And then FSH will have a very important role in stimulating spermatogenesis. And the inhibin and the actin will also have an effect, but that's kind of like a secondary effect to that. So it would be additionally influenced by inhibin and actin. Also FSH play a very important role into stimulating the Sertoli cells, stimulating the Sertoli cells. Sertoli cells are very important because they provide a lot of support for spermatogenesis. And luteinizing hormones mostly acts on the Leydig cells that actually stimulate the production of the testosterone and DHS. So that is have more role for the secondary sex characteristics. So they will have like their little nuance. So for male anatomy, I'm kind of probably not going to go through too much of it. It's pretty straightforward. I'll focus on, you can kind of go through. I highlighted something that you need to remember. And we'll work on spermatogenesis. That's a bit more, maybe it would benefit for more explanation. Okay, so the word spermatogenesis is, of course, implies that it would be production of the sperm. Leydig cells. So first the Leydig cells, this one's here in purple. They have a very important role for secreting testosterone and influence spermatogenesis. The Sertoli cells, which is right here, they nourish and develop, nourish the development. They produce a lot of different compounds that help maturation of the sperm. They also work on removing the excess cytoplasm from the sperm, because you can see how different the mature sperm is from the progenitor cells, right? There's so much more cytoplasm and so much less cytoplasm here. So the inhibin will then inhibit the FSH secretion and will inhibit spermatogenesis, and activin will actually stimulate FSH and stimulate the spermatogenesis. So the Sertoli cells will have a very important role, as I said, support, nourishment, and the Leydig cells will secrete testosterone. And DHT, sorry. Okay, so kind of a little bit of a closer look. So the first, we have the very kind of early cells they call spermatogonium, right? So they derive via mitosis here. So derive via mitosis, and then once you get to, this is like the way the reason they divide by mitosis is just to keep the spermatogonium in the good quantities, and then once they move on and differentiate into primary spermatocytes, this is when meiotic division begins. So remember, meiotic division is one DNA replication and two divisions. So we're actually going to end up with the half of the chromosomes, right? So primary spermatocytes is meiosis 1. The daughter cells of them, of the primary spermatocytes, called secondary spermatocytes, and then spermatids. Meiosis is complete. Meiosis is complete, and you have the 4 cells, and at this point, they're going to just maturate into mature sperm, where the flagellum going to develop this kind of whip-like motion that enables by flagellum, and also removing the cytoplasm that Sertoli cells play a role. See how you can see that they sort of like embedded into Sertoli cells for their final preparation for the maturation. So this one I just mentioned, and then the chromatin of nucleus has become very, very condensed and prepared for potential fertilization. The stages, you don't really have to memorize these names, but more to understand that the mitotic division is just keeping the numbers. And then once they start differentiating, it's all meiosis. Because remember, you have to half of your chromosome by the end of meiosis. So this is kind of another look on the development, on the spermatogenesis. Changes in the production of flagellum, removing excess of the cytoplasm, and finally the mature sperm. So this is a scanning microscopy of the mature sperm, so you can kind of perhaps identify the head and the flagellum. The midpiece is a little harder to see here. It's just a little on the thicker side. Right here, I can see that clearly. So this part. A lot of mitochondria to enable the motility. So here's the closer look. So the head contains DNA. So this is a nucleus, very condensed DNA. There is also an area called acrosome that has actually enzymes that will be able to penetrate the corona radiata and the protection that the ovum has to be able to actually pass through the corona radiata and then actually be able to fertilize. So these enzymes are very, very crucial for the sperm to be able to fertilize the egg. So here we have a lot of mitochondria, so that's located in what we call it meat piece. And of course, flagellum is very, very important for the movement, motility. So it takes about 64 days for the maturation. And then in the epididymis there is another little additional nutrients, we'll look at them in a minute, that will actually finally mature. So that's another 12 to 20 days to kind of put some final touches on that. And about 200 million spermatocytes are actually generated every day. So a huge numbers there, right? So here's the also scanning microscopy where you can see the sperm is actually trying to penetrate yet another level of the ooze called zona pellucida. And when we look at the ovum and the fertilization, it's kind of will be a little bit more clear. But this is a scanning microscopy, so that's not the artistic rendering. So you can see that very well. Usually after ejaculation, we have outside of the body, so 24, 48 hours, and then 5 days inside of anybody's body, I guess. Doesn't have to be a woman's body. Cryopreservation actually does work. There is as you guys know, sperm banks we have. The temperature is minus 90 centigrade. It's a liquid nitrogen. Sometimes people go through the cancer treatment, right? Vasectomy could be different risk of death if people preserve their sperm or the slow sperm count exchange operation. All that could warrant cryopreservation of the sperm. And we do know that we do have cases where the 40 years success storage. I have a very good friend who's she actually didn't freeze the sperm. They froze the embryos after in vitro fertilization because they kind of didn't think they're going to have any more child. They had the first one by in vitro fertilization. So those are embryos, meaning already fertilized, and they usually let them, divvy up to a few cells, 10 to 100 cells, and at that point, they could be preserved, cryopreserved. So she did that, and then like 6 years later, they decided to throw them out, and so they have twins. Because what of course happens is when they put multiple embryos, because they don't know the viability, how many will survive after 6 years in the freezer. So yeah, those kids are going to college now. So I always look at them, and I'm always looking at them thinking, they were in the freezer for 6 years. So science, right? Fantastic. So, as we know, lower temperature is required for sperm production, lower than the core, right? And that's why we have a descent of the testis, right? So this is very important. And actually, increasing temperature, even 1 degree, will actually result in 80% reduction of spermatogenesis. So one of the things that people do, of course, is like if somebody's having low sperm count, is suggest to make sure they not in a situation where they're overheating, or maybe very tight clothing, or spending too much time in the sauna. They're trying to conceive a child, right? Tight clothing. So the colder the temperature, well, not colder, so 2 to 3 degree lower than the body temperature would actually promote spermatogenesis. So this slide has both estradiol and the testosterone, but we're focusing on the males right now. So testosterone is in blue, and these little squares and the solid lines are the testosterone in males. So even though there is a little bit of a decline, but if we compare to estradiol and the female, nothing like that, right? So just a little bit of a decline in testosterone production. So this one was the broken line, that's testosterone in females. So females, testosterone, and here's the male testosterone over the time. This little peak here is really, well, first of all, there is another peak over here, which is not a very good slide. That is the important for the testis development during embryonic development. That's that. I know it's kind of hard to say, but see that little before birth, and there is a little bit of a blue line there. So the high level during embryonic development, that's for the gender progression. Then it kind of drops down below, and then we have that little second surge of testosterone. Sometimes they call it like mini puberty. Very important for the brain development. So that's another little spike. Nothing, of course, like during the teenage years, but significant. And, and a slow reduction that we do call the andropause. DHT and testosterone, although both androgens, they do have a little bit of a differences in how they affect male. So for the secondary sex characteristics, the testosterone is important for external genitalia, testosterone and DHT. Sperm production both, kind of similar here, but libido and the sex drive, that's testosterone. It has an anabolic effect, meaning it stimulates the protein system, muscle growth, bone growth, and therefore, usually on average, male do present larger, right? It has an effect on the decrease in body fat. Baldness and hair growth is associated more with the DHT rather than the testosterone. Erythropoietin production, more red blood cells in the male. Remember, we were looking at hematocrit values, the males were higher values, right? That's actually influenced by testosterone as well. It could help with the depression, but it also has an effect of more aggressive behavior, and of course, negative feedback to gonadotropin and LH. Seminal fluid has a lot of important function to lubricate, to provide the energy. So for example, the mucus for lubrication, and the bulboureteral glands are what produce the mucus. Water, of course, is to provide the medium for the sperm to be at. There is a multiple buffers produced by prostate and bulboureteral glands as well, and they're very important because normally vagina environment is acidic, and this will be not good for the motility. So this neutralization is important. So there is a multiple nutrients such as energy supplies and other like the vitamin C and citric acid is very important into keeping the capillaries healthy. So seminal vesicles, prostate, epididymis, they all kind of supply all these different nutrients. That's very important for the sperm. Enzyme that, because what happens sometimes is that during the insemination, right, the way to kind of liquefy the clot in the vagina sometimes presents, especially to avoid multiple fertilization. So there's a multiple enzymes produced in the seminal fluid. Zinc has a lot of different publication lately. I don't think they're quite sure about that. There's some correlation with the fertility. The zinc is also present in the seminal fluid, and the prostaglandin that help with the muscle contraction and the muscle contraction, the kind of the transportation of the sperm. So, as we know, parasympathetic neuron inhibit vasoconstriction. And so when they inhibit vasoconstriction, that of course help with the erection, right? So, but sympathetic nervous system is also very important because it increases, because remember those are the parasympathetic and sympathetic, they kind of work in concert for the erection and ejaculation, because it increases the production of nitric oxide. And nitric oxide is very important for maintaining the erection. So also prevents sperm from entering the bladder. The REM sleep, most of you of course heard of that. It is non-sexual erection. It could happen several times a night, usually during the young age. For the dysfunction or potential kind of a problem, so erectile dysfunction is inability to sustain erection, called AD. The impotence is inability to conceive a child after 1 year of trying unprotected sex. So after 1 year, usually at that point, it could be considered impotence. Excuse me, what did I say? Infertility, that's what I wanted to say.
Yeah.
I was like, why am I saying impotent? Doesn't make any sense. Infertility, erectile dysfunction or impotence, that's an ability to maintain erection. Gosh, infertility is an ability to conceive a child after 1 year of trying. So multiple, of course, reason for that. But what I've seen in the literature that kind of interesting, that sometimes in men over 40, the ED could be a hallmark is actually cardiovascular disease. It could be the first hallmark of cardiovascular disease and atherosclerosis. Because cardiovascular disease and atherosclerosis will be actually influencing the ability to sustain erection. And sometimes that is actually the first sign. And so then they have to go see what's happening with their cardiovascular system. And also with atherosclerotic and the diabetes, they inhibit ability of the endothelial cells produce nitric oxide, and the nitric oxide is very good vasodilator. So that altogether could affect the ED. Alcohol usually inhibits sexual performance, however, it does increase the desire. So you have to be balancing that out, right? Just stop at the right time. So here's infertility, that's what I was gonna say, and then I was saying something, duh. So after 1 year. So what could be a reason? Low sperm count, of course, the sperm that is not, not developing correctly. So some kind of a sports or activity that maybe keeping the testes in the high temperature, that could dramatically decrease the low count. Sometimes I see reports on professional cyclists, so they have to take extra measures to make sure they keep it cool. For the females, any problem with the fallopian, other structural problem, the lack or decrease of ovulation, of course. There are actually reports also of the females producing antibodies against the partner's sperm. Yeah, treating them as a foreign antigen. So that is a very serious actually issue. And a third of pregnancy usually terminates without kind of anybody knows. Self-abort, because it could be different situation in chromosomal count or any other problems that you would really know you were pregnant. Okay, I did mention disjunction. Why am I bringing this up here? Sorry guys, I feel like we already did that. Did I just double the slides? Okay. So that's a part one. What I was